Today we are going to finish up understanding how glucose oxidation gives us ATP by talking about the respiratory chain, which happens in the mitochondria and is really the origin of this phrase, which has now become a meme. So we've already talked now about the stage 1, which was glycolysis, and then that transition reaction, stage 2, which was the TCA cycle. So like I said, here we need to talk about how all those NADHs and the 1-FADH2 that we took out from the citric acid cycle can create ATP while using oxygen. So all of this takes place in the mitochondria. So just to kind of go over what we need to know about the mitochondria, they are, of course, an organelle found in eukaryotic cells. They have two membranes, an outer, very porous membrane that most small molecules and ions can pass through, even if they're polar. And then it has an inner membrane that is much more impermeable, and you need more transporters and things to go across it. So the inner membrane is where the respiratory complex we're going to talk about here is. It's where ATP synthase is. Those are membrane proteins. The liquid phase on the inside of the mitochondria is called the matrix. And the matrix also contains many important complexes that we've talked about that are soluble, such as the pyruvate dehydrogenase complex, the enzymes of the citric acid cycle, and once we get there, they also contain fatty acid oxidation and amino acid oxidation enzymes. So the only thing that we have talked about so far with oxidation is that it does not take place in the mitochondria has been glycolysis. Everything else, once we get past, so glycolysis does actually take place in the cytosol, but once we get past that, then the pyruvate moves into the mitochondria, and then everything else happens in here. So that is why it's called the powerhouse of the cell, because it's where we're producing all of that ATP for us. Mitochondria have their own genetic makeup. They have their own genome. It is very small, and it mostly just contains the genes for things like these enzymes, the citric acid cycle, the respiratory chain, that sort of thing. So today we're focused on the respiratory chain, and it has 5 important cofactors that can be oxidized and reduced that can carry electrons. 2 of these we've, of course, talked already at length about, NAD plus or H, and then FAD or FADH2. Another 2 that we've talked about, but not really in this terms, so ubiquinone or coenzyme Q. We talked about this when we talked about fats, because this ubiquinone is a terpene, these long isoprene chains. But we haven't talked about how it works to be a redox reagent. And heme, which we talked about, of course, at length with hemoglobin and myoglobin, but again, we haven't talked about how that could affect redox reactions. And then one we haven't talked about, but it's relatively simple, and this is just an iron-sulfur center. And these come in various different varieties, but the gist here is that we have iron again, and it's usually bound to some sulfurs from side chains of amino acids and sometimes other sulfurs as well. And in heme and iron-sulfur centers, really what the focus here is the iron. So we're going to talk about those other 3 just so we can talk about how they deal with the electrons. So ubiquinone, which we can also just call Q for ubiquinone. And so this can accept 2 electrons. It does pick up 1 at a time. So it goes, here's the fully oxidized form, which would be Q. So without the H's, it's fully oxidized. And then it picks up a hydrogen atom, so a proton plus 1 electron. And that becomes a semiquinone radical, QH. And usually, actually, we're going to see that it then picks up another hydrogen atom to become ubiquinol, which is the fully reduced form, QH2. So what we're mostly going to talk about is Q to QH2. Right, we should say plus 2 hydrogens over here. Again, these hydrogens can come from various places, but we're not implying where they come from. So I don't worry about the semiquinone radical. You should definitely know that this is the oxidized form and this is the reduced form. The hemes can accept only one electron at a time. It can accept one and then give it up and accept one and give it up. And that goes on to the iron. And this is just because iron can come, it can be in either the 2+ or the 3+ oxidation state relatively happily. And so we can, if you're in the 3+, you can pick up one electron and become 2+, but you can't pick up another one. You have to give that one back off and then pick up another one. So this is just, there are various different types of hemes, and it doesn't change the iron. That's all that's happening, but there might be different stuff sticking off of this basic porphyrin ring. Proteins that bind to heme and use it for its redox properties, as opposed to, say, hemoglobin, are called cytochromes. And so when you see cytochrome, you should assume that the important cofactor that's binding to the electrons is going to be some sort of heme. Iron sulfur centers then can accept one electron per iron, but some iron sulfur centers are going to have more than one iron. So this one, for instance, could accept 2 electrons, 1 on each iron, and this one could actually accept 4. We're not going to focus too much on iron sulfur centers. You should know that they exist, but we're going to focus a little bit more really on the Q and the other ones we've already seen. All right, so the respiratory chain has 4 complexes, and what this is showing you is that one way we can tell that is you can take the mitochondria, you can break it apart, you can then use detergent to get all of the integral membrane proteins out of the membrane, and you can then run it through ion exchange chromatography to separate out all of the proteins. And if you then, you're going to get more than that, but the 5 ones that are important here, if you treat them with different reagents, you're going to figure out that one of them can convert NADH to Q, one of them converts succinate to Q, one converts Q to site C, the other one converts site C to oxygen, and the final one makes ATP. The respiratory chain is really just considered to be these first 4, and then ATP synthesis is sort of its own thing. But however you think about it, we've got these 4 complexes, and then we have ATP synthase. So we're just going to talk about these individually. So the first complex, complex 1, is called NADH dehydrogenase, which should give us an idea of what's happening. This is going to convert NADH back to the oxidized form, NAD+. And I want to point out there's another error here in this figure. This arrow is just going the wrong direction. So if we are oxidizing NADH, we obviously have to reduce something. And the final molecule that's going to get reduced is Q. So we're going to go from Q to QH2. And in the process of doing that, we are going to also move 4 protons across the membrane. This 5th proton here is because Q picks up both of the protons when NADH only had one of them. So 1 proton extra is going to go on to here along with the 2 electrons from NADH. And then the other 4 are just simply going to get moved through. This does not happen directly. It happens through a series. The electrons are actually passed through a series of electron carriers, including FMN, which is a variant of FNAD or FAD, a number of iron sulfur centers, and eventually moved on to the Q. But you can really think about it as being this overall reaction. And so I want to point out this N and P. So that stands for the negative and positive sides. So the matrix is more negative, and the intermembrane space is more positive. When we're talking about the membrane potential, this is now not a cell anymore, so the membrane potential isn't that standard negative 0.05, but the matrix is more negative, and the intermembrane space is more positive. So that means we are using energy to move these protons across because the protons do not really want to go across. That is against their gradient. It's a positive delta G to move these protons. So this is an active transport type situation. And most of the time when we saw active transport, we were using ATP. But here the energy, instead of coming from ATP cleavage, is coming from this redox reaction. So what you need to know for sure is it recycles NADH back to NAD+. It takes Q to QH2. and it moves 4 protons across the membrane from the inside to the outside. All right, complex 2 is called succinate dehydrogenase, and I didn't really focus on this when we talked about this, but the name might sound familiar, and that's because this is one of the enzymes in the TCA cycle. It is, in fact, the enzyme from step 6, which is the step that makes FADH2. So what I didn't really make a point of at that point is FADH2 is stuck on its enzyme. It can't leave. NADH can float off and move over to complex 1 and do its thing, but FADH2 cannot. So actually what's happening is this is both an enzyme in the TCA cycle as well as an enzyme in the respiratory chain. We could have easily just said actually that that enzyme, instead of saying it made FADH2, we could say that it actually made QH2, but that's just not how it's normally said, so that's why I didn't do that. But so what this is doing is this is just another component of that enzyme, and it takes the FADH2 that was just created, and it's going to reoxidize it, and it's going to reduce Q to QH2. And again, it's going to go through a series of iron sulfur centers and whatnot, but that's going to be the gist of it. All right, and then now the FAD can react again with succinate to do this step of the TCA cycle. So Q, I should maybe point out, QH2 can also leave where it is and float off and go somewhere else. So that's critical. FADH2 could not. Complex 3 now takes the ubiquinone that came from either complex 1 or complex 2. No, not both. So nothing's going to go through both complex 1 and 2. It's going to go through one or the other. And this is called the ubiquinone cytochrome C oxidoreductase. This is another sign that you're talking about a redox reaction. And this is just going to now take Q and reoxidize it. And then it's going to take a cytochrome C and reduce it. Cytochrome C is a little harder because it's just the electrons that move. Protons do not come with it, right? And because it's just the electron moving on to the iron, and so it's a little harder to indicate whether or not it's oxidized or reduced. So generally I just indicate that by oxidized or reduced. So it's a little easier to tell. While that is happening, we are going to move some protons into, again, outside. So we're going to move 4 to the outside. 2 of those are going to be the protons that are left over when the electrons move off of QH2 and onto the cytochrome. And the other 2 are just going to be protons that were in here. So these, because there are 2 electrons in each QH2, but a cytochrome can only pick up 1, this has to be a 1-to-2 ratio. So again, there's a lot going on these figures, and I really think it's way more than you need to focus on. But so maybe take what's necessary for the figures and don't worry about the rest of it. Complex 4 is now cytochrome oxidase. It's going to take those cytochromes that were just made in complex 3 and it is going to reoxidize those. And this final step is going to take oxygen and reduce it to water. So I want to point out these numbers here. So we only created 2 reduced cytochrome Cs in the last one, and here they're pointing out that so we don't have to have halves or anything here. You do need 4 cytochromes in order to reduce 1 molecule of O2. But if we wanted to keep everything equivalent, we should really have made this 2, 4, 1/2. 2, 2, and 1. All right, so if you wanted to make this, we were moving through with either 1 NADH or 1 FADH2. You cannot reduce an entire oxygen molecule with just 1 NADH. So to keep the numbers similar. So what we're doing here then is we are actually going to use some of the protons that are in here in the N side to make that water, but we are also going to move again, some protons across the gradient. Again, here they're showing 4, but comparatively for NADH2 or NADH, this would just be 2. All right. So I think the way your book does it in this particular figure is a little confusing, but it's the way the book does it. You're looking at the book, so I don't want to confuse you too much. And this is the final step because now we've just made water and we've used the oxygen that you breathed in. So this is the final bit. So overall, and this figure I think is better because the ratios are correct here. Overall, if we have oxidation of a single NADH molecule, we're going to pump 4 protons out through complex 1. We're going to make a QH2. We're going to completely skip over complex 2. Then we're going to move 4 protons out from complex 3. And then we're going to move over here to complex 4, and we're going to move 2 protons out. So overall, 10 protons. All right, that is going to be important in just a minute when we start figuring out how this relates to ATP. FADH2, on the other hand, doesn't move as many out because it skips complex 1 entirely. It uses complex 2, and then it goes in complex 3, and it moves out these 4 and these 2, but so in total it only moves 6 out. So we don't get as much energy out of FADH2 oxidation, and therefore we cannot move as many protons across. So this is making a proton gradient, and so that uses the energy from this NADH or the FADH2 oxidation. So if we just look at really the final overall, right, all those Qs and the cytochromes, those all got recycled, but the overall was NADH plus H plus half an oxygen goes to NAD plus water. So the delta G of that, we could calculate this from the redox potentials, and that would be negative 220 kilojoules per mole. So that's a pretty good amount of energy. The energy that it takes to move 10 protons across the gradient is we could also calculate this with, this is a variant of the one we used in chapter 11 to talk about gradients. This is a little bit different because since it's protons, we can talk about pH versus concentration, but it's the same idea. And that comes out to be about 200 kilojoules per mole. So essentially, we use 200 of this negative 220 that we make, and we use that to move these protons across. And we leave a few, that extra 20 kilojoules per mole is so that the overall thing stays favorable. So we spent all that energy moving them out, and the entire purpose of that was so that we could let them flow back in through ATP synthase, which would be with its gradient and therefore favorable. So this is kind of like if you could pump water up a hill and then you let it flow back down through sort of a water wheel, and that maybe use that water wheel to turn something and grind some grain or something, whatever one does with water wheels, right? You would be getting energy out because you're letting the water fall back down, but it does take energy to move it up in the first place. And this can be useful if, it's different types of energy that you need. So the overall reaction here is going to be that ADP plus phosphate is going to form an ATP and water, but we're also going to have to move some protons from the positive side to the negative side to make this favorable. And right now I'm not telling you the number because it is slightly going to vary depending on what we're going to talk about next. So we've seen this ATP synthase before. It's an F-type proton pump that does not, the proton pump would be using energy from ATP cleavage to move protons against a gradient. This is doing the reverse. So the way it does this is that the problem here with trying to make delta G, right, is we have a very high activation energy and we also have a very positive delta G, right? It's about positive 30.5 kilojoules per mole. That's a really big number, not very favorable. So what this enzyme does is it changes things so that the creation of ATP is not high anymore. It's the letting go of it. And so here we have ADP binds, or ADP and phosphate, and then those bind to the enzyme, and that's going to have a slightly negative delta G. And then because of the way the enzyme is because of the structure of the enzyme, it actually then has a slightly negative delta G to make ATP as long as it is bound to the enzyme. And then the really big, but we cannot change the fact that this has to eventually be the same delta G. But the way it deals with it is this huge high thing is the energy of it falling off. So the high energy does not occur until it tries to let go of the ATP. And that means that we can physically force it off. So the way this works is we have many different types of domains in the ATP synthase. The ones I want to call your attention to is the beta domain out here, and this is on the N side, which remember is the inside. This is the matrix side of the mitochondria. And so this has 3 beta domains interspersed with alpha domains, but the beta domain is where the actual reaction happens. And then the other thing I want to call your attention to is the C domains. And so this is one C domain. And there are a different number of these, and they make up a ring that are actually in the membrane. And these two are linked by the gamma domain, and when this turns, so it's like an axle of a car where they both have to turn at the same time, right? So protons are going to come in, so remember we have more protons on the P side. They're going to move in here, bind to a C domain, and then they're going to sort of rotate around. And then when they get to the other side, at the very end, then they're going to pop out. But they can't come out until they've gone all the way around. And so every time, it's going to take however many numbers of C units we have, that's how many turns it has to make, how many times it has to move to turn all the way around. There's always going to be 3 beta domains. And so one complete circle of this is going to also lead to one complete circle of this. So the way this works is there are these 3 beta subunits, and they have 3 different conformations that preferentially bind to one or of something or nothing. So one conformation is where it's ADP has a very high affinity for it. So this subunit is when ADP binds. When it moves over here, it changes conformation slightly, and that is when it becomes favorable for ADP to react with phosphate and then to become ATP. But this domain does not want to, it has changed configuration so that it doesn't want to hold on to ATP anymore. So when it shifts around here, the ATP falls off. And we can force it into this configuration by turning this wheel. And what's causing the wheel to turn is the movement of the C domains with the protons in them. So, like I said, there's going to be 1 proton bound to each C domain, and it wants to turn because every time it turns, it can pick up 1 proton from the P side and drop 1 proton off in the N side. And that's where the protons want to go. So the number we're generally going to use is an average of 9. And that number actually comes from an average of different eukaryotes, but the 9 is also actually a particularly useful number because, as you'll see, it gives us an even number. So actually that's probably the real reason we use 9 as opposed to using the number that, say, humans have. So we're going to assume that for the beta subunits to turn an entire turn, we're going to have to move 9 protons through the protein with the gradient. So we also want to point out that the full rotation of the top subunits is going to make 3 ATPs. And that's because if you took a snapshot, we don't start with nothing on it, right? We start maybe here at any given point where there's already an ADP on here, there's already an ATP on here, and there's already an empty spot. So the full rotation, each subunit is going to go through all 3 and is going to make 3 ATPs. So for every 9 protons, that goes through, we're going to make 3 ATPs, and we can consider that essentially for each ATP, it took 3 protons to turn it 1/3 of the way. One other thing to point out is something else that has to happen. All this ADP that we want to turn into ATP is probably not in the matrix. We used it all. We used all the ATP out in the cytosol. So we have to move the ADP into the matrix, and then we also have to move the ATP back out so that it can be used again out in the cytosol. And the gradient makes this favorable because we are moving out something that is a little more negative. So this is a cotransporter that moves in one ADP and moves out one ATP. ATP is a little more negative than ADP, however, so this is favorable because we're moving the more negative thing out. There's also a phosphate translocase, which moves a phosphate in. This is the form that phosphate is generally in at pH 7, and that would be against the gradient. Right? We'd be bringing a negative thing in to where it's already negative, and that would be bad. So it also brings in a proton. So it's another cotransporter. This is a symporter. Brings in another proton, and that's for every ATP that gets made, right? So every ATP that gets made, we're going to bring in 3 of these protons over here. We're going to bring a 4th one over here to get the phosphate in, and so that is actually a critical proton right here. So we have ADP plus phosphate plus 4 protons moving in makes ATP. So now if we think about how does that relate to the number of protons that we moved out with either NADH or FADH2. So one NADH moved out 10 protons, and so we can make more than one ATP with that. So just 10 divided by 4 means that we can make 2.5 ATP per NADH. FADH2 didn't move out as many, we only moved out 6, so we only get 1.5 ATP per FADH2. These are absolutely numbers you should just know. I mean, you could try to go back and rederive this, but I wouldn't unless I asked you to do something funny about it, right? I would absolutely just know these numbers off the top of your head. So now we can talk about how much ATP we can get from glucose. And we can also see that most of it comes from respiration because we get so much ATP out of these NADs and FADs, H2s. Right, so we know that glycolysis, we made 2 ATPs directly. That was out in the cytosol, that's fine. They're already ready for use. But we also made 2 NADHs, which we now know we can eventually, through the respiratory chain, make 5 ATPs. So overall, glycolysis plus recycling gives us 7. That transition reaction, the oxidation of pyruvate, gives us 1 NADH. However, we actually did it twice for each glucose because we had 2 pyruvates from each glucose, so that's where this times 2 comes from. So if each NADH gives us 2.5, times 2 is 5. The TCA cycle, we got, for going through the TCA cycle 1 time, so 1 acetyl-CoA, we took 1 GTP, which we can convert into an ATP. We made 3 NADHs and an FADH2, which give us 7.5 or 1 plus 1.5. So 1 round of the TCA cycle gives us 10 ATPs. That is also a number I would absolutely just know cold off the top of your head. You should pretty quickly be able to rederive this, but it's just such a simple number. It's so nice and round, and it just will simplify things if you know that the TCA, 1 round of the TCA cycle gives you 10 ATPs eventually, once you recycle everything. And then this times 2 is because, remember, originally from 1 glucose molecule, we would have made 2 pyruvates, we would have made 2 acetyl-CoAs, so we actually have to go through TCA cycle twice, so we actually get 20. So total, if we add all these up, the 7 plus the 5 plus the 20, we end up with the total number is 32 ATPs. And of those 32, 29 of them came from the recycling of NADH and FADH2, right? Because the only 3 that didn't were these 2 from glycolysis and then this 1 that we made 28. I forgot to multiply it times 2, right? So these 2 from glycolysis and then the 2 GTPs that we made in the 2 rounds of the TCA. So 28 out of 32 came from respiration. That is the vast majority. So that is the end of glucose oxidation. And the final thing we'll be doing is talking about how this relates to fat, and that's going to be a lot simpler because it's still going to use the TCA cycle and this respiratory chain, but we've already talked about it. So that will be the last lecture that we have for next time.